Okay, you want to start us off then?
Sure, I will.
Okay, great, thanks.
People are drifting in, but we need to stay on schedule. So, if everybody could slowly digest your lunch, and we have now adjusted the clock, so the speakers, instead of having an hour and a half, they have about a little over half an hour. So let's like to get going. It's a really low. Only one. Oh, only one. How well. So, anyway, it's now 1:30 and a little after. So it's really a great pleasure to introduce Pete Homyak. Pete was born in Colombia, and he moved to the U.S. to attend college at SUNY Binghamton and graduated there in 2004. He has a master's degree from SUNY Syracuse in Forest and Natural Resources Management and a PhD in soil water science from University of California, Riverside. He postdoced for about 5 years at UC campuses, mostly at UC Santa Barbara, where he worked with Josh Schimmel and Oliver Chadwick, two microbial and soil scientists. Well, I've talked with Pete a little bit, and one of the things that many of us deal with is a dual career. So he postdoced for 5 years so that he and his wife could have similar positions at Riverside. So they met that challenge, and Pete is now an associate or an assistant professor at the UC Riverside. He has 10 first author publications, 14 total publications. I think it's a really impressive record, not only the number of publications, but the content. And Pete studies carbon, nitrogen, phosphorus, and other element cycles, movements across the soil, water boundary, soil atmosphere boundary from a microbial perspective. That's a lot of what I do, so I'm going to shut myself up right now and listen to Pete so I can learn a lot. So thank you, Pete.
Thank you so much for the invitation. It's a pleasure to be here. Actually, it's my second time here on campus. The first time I was here because I was invited for the next Professor of Science. And I think that was the first time that you had that workshop running. And so thank you so much for that, because I was able to be a good negotiator when I finally got a faculty offer. They were pretty impressed. So I did all the tough and the good stuff. So today what I want to talk about is basically talk a little bit about drought and how it controls ecosystem nitrogen cycling, and basically challenge established paradigms of how we think drought controls ecosystem nitrogen cycling, and argue that important microscale as well as microbial processes can actually influence the cycling of nitrogen during a drought. Before I do that, I would like to thank many of the people who made all this work possible. That includes the Schimmel lab at UCSB, as well as the Allison, Treseder, Golden, and Hooksman labs at UCI, and then many undergraduate students who really made everything that I'm going to show today possible. So first, I would like to start by convincing you that Earth is a dry place. That's because drylands comprise about 40% of the land surface, highlighted here in yellow. And even in these regions where they're not highlighted in yellow, we can actually argue that they experience drought one way or another. In other words, if we look at the percent land area and we look at the number of dry months, we notice that about only 5% of the land area is this always wet, and those are those blue dots. The rest of the map will see drought again for at least 1, 2, or 3 months, or then potentially all year round. And so if we look at these ecosystems and we think about how nitrogen cycling happens in these systems, theory tells us that these systems are extremely nitrogen limited. That is because there is not much water, and so processes like decomposition and processes in terms of interactions between microbes and plants are stressed. And if you stress those mechanisms, then it's perhaps easy to predict that these systems will have very little nitrogen to go around. Interestingly, though, these are also systems that during certain time periods, paradoxically, just lose tons and tons of nitrogen. They lose nitrogen as if they were nitrogen saturated. In other words, as if they had so much nitrogen that they wouldn't care to lose some during specific time periods. And those time periods actually happen when the rains return. So for instance, in California, we go about 6 months without rain, and then suddenly rain comes back. This year has been a tremendous wet year. And during those time periods, we can lose nitrogen from these systems through hydrologic pathways, for instance, nitrate. But what I want to talk today is going to be the loss through these gaseous pathways, and in particular, to focus on nitric oxide. And the reason I want to focus on nitric oxide is for two reasons. One of them is that it actually regulates the oxidative capacity of the atmosphere. And so if we think of nitric oxide, it actually participates in the production of hydroxyl radical. And we need hydroxyl radical because it's a cleanser of pollutants. So in a sense, nitric oxide can be good. But if we have nitric oxide in excess, we can actually catalyze the production of tropospheric ozone. And what I want to show here is a picture I took on Sequoia National Park. This is a system that we think it's remote, that's isolated from human activity, and that everything should be pristine. But in fact, you can see this really nice air pollution layer where these systems are actually severely impacted by ozone as well as nitrogen deposition. And so this then begs the question that we must understand how drought controls the mechanisms that release these gases, especially from these natural ecosystems. So with that then, what I would like to do with this talk is just answer a simple question. How does drought control ecosystem nitrogen loss? So again, when we think about the dry season, perhaps these are pictures that will come to mind. Everything seems to be dead. Deer struggle to find water. And theory, again, tells us that ecosystem processes shut down and that microbes will die because of diffusion constraints. But I'll argue that if we actually try to look in the literature specifically for how these microbial processes shut down and that the nitrogen cycling shuts down, we actually don't find much support for it in the sense of looking at this metric. So what I did was I searched the literature for things like nitrogen mineralization, nitrification, gross mineralization, and I compiled all those measurements into a simple metric that I called nitrogen supply. And with that meta-analysis, I showed that as soil moisture reduction increases, in other words, we're making things drier as we go to the right, we really don't see much in terms of an effect. In fact, if you look at the entire average, it actually sits right about 0, meaning that we just really cannot predict either a positive effect or a negative effect of drought. In other words, it seems as if nitrogen supply is sustained even when systems dry out. And that is actually the key of my talk, that sustained nitrogen supply. Because when we think about nitrogen cycling, it helps to look at it from this cartoon. And so such that if you are a bacteria that processes nitrogen, either ammonia oxidizer or in particular this nitrite oxidizer, you're just sitting at the communal and you're just waiting and you're saying, I'm going to finish his nitrites. Because when you can finish that metabolic handoff from one set of organisms to the other, that's when you can couple the nitrogen cycle and efficiently cycle nitrogen. My point is that this interaction between all these microbes at the communal, it can potentially be interrupted by drought. And that under those conditions, maybe we can generate pathways during which we can actually lose lots of nitrogen. In other words, that there is no efficient transfer between these leftovers, and then the leftovers go to waste. So to bring that together, we can go back to the literature and pull out this model from Mary Firestone and Eric Davidson from 1989. This hole-in-the-pipe conceptual model. And it suggests that we can envision nitrogen as flowing through a pipe. Very simply, we can look at ammonium being oxidized over to nitrite by ammonia oxidizers. Nitrite in red because I want to emphasize that that is the key metabolic handoff. And that then the nitrite can actually be oxidized by nitrite oxidizers over to nitrate. Eventually, nitrate can be denitrified to produce nitrite again. And then from there, through this hole in the pipe, we can release NO. So the key here is understanding whether nitrite is actually efficiently transferred over to other organisms and whether drought affects that transfer. So the hypothesis then for this talk is that drought concentrates reactive metabolic handoffs, generating N leaks when soils wet up once again. And to highlight that, I would like to show this image. This is representing a soil when it's wet. And so if soils are wet, there is hydrologic connectivity. All these substrates, ammonium, nitrate, nitrite, are just floating around in that soil solution. But that if we expose these soils to drought, we will remove water, and we will just leave thin water films on soil microsites. And that under those conditions, perhaps, is when we can generate nitrite accumulation. Because so it could happen that this ammonium gets oxidized over to nitrite, but there simply isn't a nitrite oxidizer to pick up that molecule of nitrite, and therefore it can only accumulate and it just sits there in the soil. So to test that, I had to go to a California annual grassland because it's the perfect system. It's a system that can actually go about 6 months without rain. And so if we want to study drought, there's nothing better than studying it right here. And I did several measurements. So I applied some landscape scale measurements to understand whether nitrite would accumulate. Then I came down to the microscale, and then I used a dual isotope approach to really understand these processes. So let's look first at the landscape scale, detecting whether we can actually detect, try to detect nitrite accumulation in soils. And long story short, this is exactly what I found, nothing. So I tried to measure whether nitrite would accumulate over the length of the dry season, and it was virtually impossible. I found refuge in reading some soil manuals that told me that nitrite is typically not found in detectable amounts in soil and therefore is usually not determined. This is what you just find if you look at nitrite in the literature. That's basically what you find. But we were stubborn, and so we actually found out that all these methods have been wrong. They're wrong, especially if you are in an acidic soil. And what happens is that you're actually blowing off the nitrite you want to measure as NO, and that's why we don't measure it. But with this new technique, then, I was able to produce that figure where we actually measured nitrite here on the Y, then time here on the x-axis, and water-filled pore space, a metric of how much water is in the soil. You notice that water-filled pore space decreases during this first dry season, and that nitrite tends to build up. And then during the second dry season, the same occurs. Nitrite tends to build up. And this was useful because it gave us kind of like the same, like the general patterns that we were expecting. But when we put stats through this, we actually find that those slopes are actually not significantly different from zero. There's just too much variation, and we really cannot demonstrate that nitrite accumulates in a dry soil. So that's when we went to the microscale approach. And we try to use a technique to see if we can detect nitrite accumulation at a microscale rather than trying to use these bulk measurements at the ecosystem scale. And we use a technique that is actually used in studies that examine the chemistry of brains in rats. And it's called microdialysis, and it's a tiny little probe, and it has a little membrane, this white little piece right there. And the membrane acts like this figure here. You can insert the membrane in the soil, and then this is the blow up of that membrane. Water comes through the middle, down this pathway, and then it returns through the outside, so that any soil in contact with that membrane will exchange solutes, and then you can pick up the solution at the other end in a tiny little microfuge tube. And we use that technique to see if we can actually detect nitrite in soils. So this is what we found. What I'm showing you here is nitric oxide flux in this top, then nitrite in the middle, and then nitrate at the bottom. And if I plot a line through that, I hope I can demonstrate that, in fact, when peak nitric oxide occurs, it seems to correspond quite well when peak nitrite concentration happens. And that when nitric oxide decreases, it coincides with the decrease in nitrite. And it also happens to coincide with the increase in nitrate. In other words, providing some support of the idea of this metabolic handoff, that the ammonia oxidizers are fast at producing this handoff, but that the nitrite oxidizers are slow, and so that when these handouts are not being consumed by the slower nitrite oxidizers, then you can generate some of those nitrogen leaks via gaseous pathways. In other words, that this breakup and handing off nitrite can lead to producing nitrogen leaks. So this microscale approach was useful. It was useful because it gave us an idea of some support to the hypothesis we had. But it was not useful in the sense that this was not really the hard evidence that we needed to demonstrate this process. And so what we did was turned over to isotopes to answer the question, what is it that controls gaseous nitrogen leaks at the landscape scale? And so under this type of scenario, we actually use an isotope tracer experiment. And we use that dual isotope tracer experiment. We focused on measuring the N15 composition and the oxygen 18 composition of nitric oxide. And we did that for two reasons. One, because we wanted to know the how, which were the processes involved in producing nitric oxide. And then we also wanted to know the when, whether those metabolic handoffs I've been talking about actually do accumulate over the dry season, or are they just a product of when soils wet up? And so, in other words, that it's just rain that comes in, and that's when things become active, how we would traditionally understand nitrogen cycling to occur. And so to understand the how, I'll first focus on N15, we added N15 labeled ammonium, N15 labeled nitrite, and N15 labeled nitrate. When we added N15 labeled ammonium, we were trying to understand whether it was simply driven by nitrification. When we added the nitrite, whether chemodenitrification would be the driver. Nitrite is extremely reactive, and it can abiotically decompose to NO in a soil. And then when we added N15 nitrate, it was to understand whether these processes were driven by denitrification. So here are the results from that study. I'm plotting the N15 of nitric oxide here on the Y, and then on the X, I have time since wet up. First, 0 to 15 minutes after adding water, then about an hour, 24 hours, and 48 hours. And you notice that, in fact, during those 0 to 15 minutes, the substrate that shows up immediately in producing nitric oxide is actually nitrite. So we did find evidence to suggest that that first pulse of emissions that occurs when a dry soil gets wet up is driven by the accumulation of nitrite. What this does not show is whether that nitrite was generated right there when we added the water or whether it actually had been accumulated over the length of the dry season. We then found out that over time, at about 24 hours, that's about the time that we would expect microbes to recover from drought stress. And in fact, we find that during that period, the major driver of these gas emissions is driven by the use of the triangle, which is ammonium. So basically, nitrification driving those processes. All right, so now we know that nitrite can be important. The question is that nitrite being driven by drought or not? And so that's when we go to the oxygen 18s. And we use just basically nature to give us that answer. We didn't have to label anything. We just used this evaporation-induced fractionation. In other words, that if you have a wet soil, we now know from previous talks that you have both oxygen 16 and oxygen 18. But that if you put that soil through evaporation, 6 months without rain in California grassland, that over those 6 months, the soil will start to look like this. And that if you had any water films left behind, the chances would be that that water will be oxygen 18 rich. It will be heavy water. And if that heavy water was actually fueling some sort of microbial processes that would produce nitrite, then that nitrite should actually have an oxygen 18. It should be relatively heavier than if it was produced here when there were more oxygen 16s around. And so we went with that assumption. And we found out that our assumption was correct. We found that oxygen 18 of nitric oxide during those 0 to 15 minutes is relatively heavier compared to those compared to 24 hours when we know that microbes recover from that drought-induced stress. So that initial flush, the first production of nitric oxide, is driven by nitrite produced during the dry season with this heavy oxygen. But that later on, that peak and the trail-off of nitrification is driven by nitrite produced post-wetting, by that lighter oxygen from that added water. And so we were able to conclusively demonstrate that these processes over drought were actually important and were generating nitrite. And so then putting all of this together, we tried to come up with an idea to kind of rationalize what happens when these ecosystems go under drought stress, and why is it that they lose nitrogen as they do, as if they were nitrogen saturated. And the idea would be that when a soil is moist, we have some nitrogen flowing through that pipe, and that the holes in that pipe are relatively small. The soils are wet, so gas diffusivity will be low, and so there will be a tendency of less nitrogen to be emitted. Not only that, but you have plants that are alive, so they're competing with microbes for nitrogen. So there will be less nitrogen to escape through those holes. Over time, when you start going into the dry season, we would argue that the holes will start to become larger because diffusivity through that soil will be higher. So gases being produced will have a higher tendency to escape. And then during wet up, the size of the pipe actually increases. We measure higher rates of nitrification and mineralization. And we also now know that this handoff, this metabolic intermediate, this nitrite, actually accumulates as well, generating large holes that can actually put cracks through the pipe, if you will. And then we have large emissions of nitric oxide. And in particular, it may also apply to nitrous oxide as well, which is a potent greenhouse gas. And so to conclude then, we found that drought actually does control the loss through these trade-offs between microbial and abiotic processes, that microbial processes do endure through drought conditions, and that they concentrate these reactive substrates that produce N losses when soils wet up. And then thinking about the future, if we think about expanding drought, which models predict that we're going to have, that drylands are going to expand into the future, and if we combine that with atmospheric nitrogen deposition, these expanding drought may actually become increasingly important in controlling nitrogen loss and affecting air quality and climate. And that's because increasing rates of atmospheric nitrogen deposition may actually select for taxa that are able to nitrify more often, as well as perhaps changes in climate that favor more of these precipitation extremes. So longer periods of drought followed by heavy precipitation extremes may actually generate the conditions that I have shown here, may generate these large pulses and losses of nitrogen from systems that are actually nitrogen limited. And with that, I'll be happy to take questions.
Questions? Does someone have the blue box? Could you use that side of the room and I'll do this side? Okay. So questions? George, let me bring.
What do you think the size of those tension water layers are when it's dry? Are they too small for microbes to live in there around the soil particles so that there's no nitrification of the nitrate? Why does it just build up instead of, as the water shrinks during the dry season, you have nitrification all along?
Right, so that's a great question. The best answer to that would be that, well, one, there are two interlinked processes in the nitrogen cycle. So we have ammonia oxidation, then we have nitrite oxidation done by a different set of bacteria. Studies suggest that nitrite oxidizers are actually more sensitive to stress than the ammonia oxidizers. And so if you think about putting these organisms through drought stress, you will tend to perhaps whack off those nitrite oxidizers a lot quicker than you would the ammonia oxidizers. So under those conditions, you would have one set of organisms that are still actively metabolizing and producing these leftovers, but because the other ones are weaker, they're not there to consume it. The question whether there will be enough water in those thin water films to sustain microbial production, the answer to that is we think that it does happen. Reason for that are perhaps two. One is the isotopes show that this nitrite is actually building up over the length of the dry season. So somehow they are able to survive on those thin water films. And then interestingly enough, when we actually measure microbial biomass carbon as an assay where we just take chloroform and we kill off whatever microbe is still alive, and when we measure that biomass over the length of the dry season, we actually see that the biomass actually increases. So the longer the soil that stay dry, the more carbon is actually tied up in microbial biomass, suggesting that they are surviving. We don't know exactly why the microbial biomass is increasing, but we think that it might have to do with top-down controls. For instance, it might not necessarily be that microbes are doing better, but perhaps that they're shielded from predators that eat them. And if you can decrease their death rates, then maybe it could be that's why the reason that microbial biomass carbon increases over time. But they do seem to hang out and they do seem to do well.
Someone over here had a question.
Oh,
Yeah.
Kind of building off your last point, you mentioned that a lot of these sort of arid climates are going to shift towards precipitation extremes, but then maybe also have different kind of precipitation intervals and maybe a switch of seasonality so that maybe precipitation doesn't match up as well with plant phenology and things like that anymore. So would you like to comment more on what kind of implications that might have for ecosystem nitrogen cycling? Like, I mean, in some ways it could be good news in terms of reducing limitation, maybe?
So in terms of like breaking that asynchrony between plant demand and these precipitation extremes, some of the work we did actually shows that if you break up that asynchrony, you tend to generate more nitric oxide. So if we end up with conditions that lessen the amount of plants that are around, that would mean that there's a higher chance actually to lose that nitrogen from the soil. So it would exacerbate the loss of it. If on the other hand, we are able to generate conditions where I don't know, for some reason the plant phenology starts to shift and we're able to keep plants longer then that may actually act to retain some of that nitrogen that otherwise is lost. But, Nate?
Do you see compensatory effects of ion deposition? So like you're seeing.
We want to record this.
Sorry, there's the possibility that you're going to lose, but then I'm thinking like the Bay Area and stuff, all the grasslands in the surrounding area are having too much nitrogen put into the system, which is increasing non-native species dominance. And so maybe there's a balance. I don't know.
Sure. I mean, if you can do a mass balance, right? And you argue that if you have more nitrogen leaving through gaseous pathways, but that same nitrogen is being deposited back from atmospheric deposition, that would meet that. The one thing I would argue is that some of these systems, so for instance, here in the East Coast, when we look at nitrogen deposition, we look at rates of, say, 8 to 10 kilograms of nitrogen per hectare per year, and everybody goes up in arms because that's too high. In these systems, some of them are receiving an order of like 100 kilograms of nitrogen per hectare per year, and those rates are huge. So yeah, I mean, under those systems, you would argue that the amount of nitrogen coming in is just so much higher than whatever may be leaving the system through gaseous pathways. So we expect overall nitrogen saturation of those systems. But I will also add, we've been measuring. Actually, I put this slide in. We've been developing this system of fast response analyzers, and we put these in the desert. And we measure two sites, one that has relatively high nitrogen deposition and one that has low nitrogen deposition. And we actually found the complete opposite. The site with the low nitrogen deposition releases more NO than the site that has nitrogen deposition, which is interesting. And at this point, we don't know how to explain it other than differences between systematic differences in sites, like things like pH, as well as there is a slight difference in the plants that occupy these spaces. And so at this point, we're just blaming plants and soils, but we don't know.
Other questions? Well, I have, there's one back there. Brendan, go ahead. Is there a cube? Thanks.
Thanks. Trying to pick apart the process. So if there's an increasing microbial biomass and maybe more available in as ammonium, then is there a way to measure whether ammonia oxidation is still occurring over the dry period and decouple that from?
Yeah, don't let my next project out in the room. That's actually where I'm heading, and I predict that that ammonia oxidation is actually quite persistent, and that the idea would be that you will continue to supply ammonium to the system under, nitrate to the system because of ammonia oxidizers being able to persist longer. The idea being that they will actually be able to access substrates even if soils are dry.
I think Brendan asked a question in a different way than I would, but could you go back a couple slides to where you had the del 18 of water or of oxygen in the, so the initial flush of del 18 NO is high, but do you have flux measurements so that you can use that isotopic signal to calculate how much NO was produced over the dry period?
We might be able to do that, but the issue here is that to make those like when we did this, we did it at the landscape scale, and so that isotope signature can actually go anywhere. We didn't contain it, and so it will be hard to estimate because a lot of it may have just been sequestered in soils, and we would miss that, and so we might attribute that to actual loss when in fact it didn't. Yeah. So the way to do that is constrain it in the chamber and then be able to do mass balances and come up with those numbers.
Yeah, that would be really interesting. So thank you. Any other questions? I think we're just about out of time, but we might have a quick one. Okay, let's thank Pete again. Thank you very much.